Eina mana, eina reo, e rau rangatira mā. Tēnā koutou, tēnā koutou katoa. Kia ora koutou, I'm Sarah Young. I'm the Dean of Science here at Waipapa Taumata Road, the University of Auckland. It's my great pleasure to welcome you here tonight and to introduce a scientist whose career has been defined by a deep-rooted passion for the understanding of the natural world, Professor Matt Templeton. Matt was born in Wellington, but having spent much of his schooling overseas, his career choices were shaped by a global perspective. After returning to Aotearoa, New Zealand, he earned both his honours degree and PhD in biochemistry from the University of Otago, laying the foundation for a remarkable career in plant pathology. Since joining the Plant Diseases Division of the DSIR in 1984, Matt has remained at the forefront of research into how microbes interact with plants, taking him from New Zealand to the Salk Institute in California and back again. His work has been instrumental in helping us understand how certain microbes evolve to become pathogens and how they manage to colonize and cause disease in plants. For the past 15 years, Matt has focused on a tiny but formidable foe, the kiwi fruit pathogen PSA, which has posed a major challenge to our horticultural sector. Matt and his team have played a key role in uncovering how this pathogen causes disease and adapts in the orchard, work that's vital, as vital as it is fascinating. Matt has also been a passionate educator for the past 20 years through his role in the Plant and Food Research Joint Graduate School since 2011. His teaching in plant microbe interactions and its importance to our bioeconomy has inspired a whole generation of students. Tonight's lecture promises to be a fascinating journey through the microscopic world of plant pathogens, how they evolve, how they operate, and why understanding them is so vital to our bioeconomy and food security. Please join me in welcoming Professor Matt Templeton.
Thanks, Sarah, for that very generous introduction. I'm really humbled by the number of people and friends, colleagues who are here. So, hopefully I do it proud. A bit nervous. So, yes, in truth, this isn't going to be a lecture so much about how pathogens cause disease in plants, but a little bit of discourse into my journey into science and teaching as well. As Sarah said, I was born in Wellington. Here it is on a reasonably nice day. I'm halfway up walking Mount Koko, which overlooks obviously the city, but also the northern suburbs where I grew up. And every time I'm in Welly, I really like wandering up there. And yeah, it's sort of a happy, nice place for me. That's as long as my plantar fasciitis isn't playing up, but yeah. So also, as Sarah said, I think there must be a spy in my computer somewhere. Went overseas. My dad worked for MFAT, and when I was 3, Dad was posted to London, and we sailed there. So I don't think there were, obviously there were commercial flights in those days, but it was probably going by ship was quicker. So I went on a boat called the Willem Rice. If you're of Dutch heritage, you or your parents may have come on this boat because its job from the sort of 50s right through to the early 70s, I think, was bringing people from the Netherlands to Aussie and New Zealand. Here I am on one of the decks. I'm looking sort of happy-ish, but I got seasick a lot, and I made this trip really memorable for my parents by throwing up pretty much every day, apparently. So, yeah. When the stabilisers came off, that was the signal to everyone. In London, where we lived for a few years, was joined by my younger brother and younger sister. Don't remember an awful lot about my education in London, although I arrived there as a natural left-hander and left writing with my right. And that's always given me a bit of a cynical view of Montessori schools, to be honest. What else? That's about it. So the rest of the time after that, came back to New Zealand for a few years, then was posted, well, my dad was posted overseas for the last 3 years of my high school, and that did leave a lasting and positive impression on my education. So Dad was posted to the United Nations. I attended the United Nations school. Just here it is on the lower east side. I had some really outstanding teachers. I think, everyone, whether a scientist or not, often looks back and sees that, they had influential teachers who really sort of shaped their interest and, later on career. So my first teacher was Gary Fletcher. He was from Moose Jaw, Saskatchewan. So probably found the culture shock coming to New York as challenging as I did. He's written Good Luck, so he obviously thought I needed it. And he was right, and fortunately I got it. So Mr. Fenn, also an excellent biology teacher, a little more stern and serious than the other teachers. I remember opening the yearbook and was quite surprised to see him smiling. But the person who possibly had the biggest influence was Brian Kahn. Amazing teacher. All his classes were just riveting. I was just completely blown away by the way he taught and all that sort of stuff. Now, if you look at this, see if anyone recognises this formula he's put in. So one of the things he was able to do was Columbia University at that stage had an outreach for schools where schools or students could receive various mutant fruit flies and do crosses and work out linkage and genetic distance. So I can't for the life of me remember what this blinking formula is, but it'll be something to do with working out. Yeah, so he was influential, but, it was still, yeah. Yeah, so I remember taking these blinking flies on holiday because I had relatives in Minnesota. They weren't so impressed by these little packets of fruit flies. Here was I making them eat this awful artificial diet and arranging all these marriages for them. So finished there and then went to Otago Uni, gravitated towards biochemistry. At the time it still is a strong department. It was a very strong department then. It was George Peterson, Warren Tate, and they, all the new stuff coming in was, brought into undergrad lectures, and it was great. There was a lot happening then. Sequencing was just being developed. I remember when introns were discovered. It was just completely mind-blowing, these sorts of things. So I then did my PhD honours and PhD with Pat Sullivan and Max Shepherd. Their lab at the time was working primarily on Candida albicans. Human pathogen didn't kind of spin my wheels so much, but Max had a friend, Robin Mitchell, at the Horticulture Division of DSIR in Auckland at the time. And so my PhD project was related to some work he did. He was a natural product chemist and was interested in toxins produced by plant pathogens. But there was the more interesting biochemistry there, which he wasn't so much able to do. So my project was looking at the mode of action of one of these toxins. Amusingly, I went through my whole undergraduate without doing any second or third year courses in topics that could be useful, like microbiology, plant science. To be fair, with Kevin Farndon, there was a lot of good plant biochemistry, so at least I got access to a whole lot of plant science. What was I going to say? Doesn't matter. Then amazingly, a whole series of jobs came up at the then Plant Diseases Division of the DSIR. They'd had a number of retirements, and so quite a few positions were available, and they particularly wanted to make some hires for people looking more at sort of interactions with between microbes and plants rather than just sort of pure pathology background. And so, I was really fortunate, having been a trained biochemist, that I was able to apply and got one of those jobs. It's one of those things where a lot of science and careers are being at the right place at the right time, and it's very lucky for me. Who knows what would have happened if those jobs weren't available or whatever. So yeah, it's fantastic. Took me a little while to sort of find my feet there, although they wanted to get into this area, wanted sort of a biochemical lens on some of these diseases. Didn't have the projects really sort of sorted for me. So the first year or so was a bit tricky. But I managed to get a position at the Salk Institute after a couple of years with Professor Chris Lamb. Got a picture of the Salk here. It doesn't do it justice really, because it's sort of looking a little bit utilitarian, but it was architecturally designed, and that their sort of main aim was that there was a pine forest right at the front of the institute. So you'd walk through this pine forest, and then you'd come through to these buildings which directed you right through to the Pacific. So that was the sort of architect's aim. The institute expanded about, 15, 20 years ago, and they chainsawed all the pine trees and built the extra labs there, much to the chagrin of the original architects. For me, this really was firstly gave me a great introduction to molecular biology, remembering really that my training in biochemistry was more with proteins rather than nucleic acids. So it was great to be at a sort of real cutting edge plant lab. The other fellow postdocs that I'm still really friendly with. So yeah, it was a super good opportunity. And of course, Francis Crick was a fellow there, and I remember going to one of his lectures. Don't remember anything he said, but he was more into the brain then. He kept cracking a joke that he had, quit molecular biology in 1958 or something. So when I went back to DSIR at Mount Albert, I was in a better place to sort of work out what I wanted to do. And the director at that time wanted to set up a group on post-harvest pathogens, so diseases of fruit after they've been picked, and worked on this particular fungus, Glomerella cingulata. I think it was an isolate U found, yeah. And in this time, I made some important hires, Eric first, and then Joe Bowen. And for a number of years, small number of years, sort of tried to tame this fungus, got a transformation system going, worked out how to do gene knockouts, picked something that seemed to be appropriate given it's a fruit rotting pathogen. I'm sure you guys have seen this in your fruit bowl. It's sort of characteristic circles here with orangey spores. for sure you've had it in your fruit bowl. And we were looking at the cell wall degrading enzymes. And as I said, we'd put together a reasonably good set of papers, more or less tamed the fungus. But in this time, we went through a transition where we were initially sort of funded by the organisation, but then the foundation was developed. So basically all that money went to Forced, you had to reapply for it. And although we got grants initially, it was getting harder and harder to just tick all the boxes they wanted, even though this was sort of seen as an important area, at least by the DSIR director at that time. It just really wasn't big enough and important enough to. So we sort of strategically changed to another fungus at that point. But before I get to that, I want to talk about a couple of projects. Completely lost track of time now, but anyway. A couple of projects which were sort of diversions, but weren't. So I'm going to talk just for a little bit on this amazing set of proteins called hydrophobins. They're unique to fungi, and they're really the essence of what makes a fungus a fungus. They help spores disperse, so they're able to coat spores with this very hydrophobic layer that makes them airborne. They help them adhere to hydrophobic surfaces like the surface of a plant or an insect, so they're really important in disease. And they're also crucial for how fungi or macrofungi develop their fruiting bodies. So without them, I think fungi would just be a sort of a homogeneous mass of mycelia, and they wouldn't be of any interest at all. So this project was started by the late Ross Beaver, a really inspirational person, fantastic scientist. One of the projects he had been working on was, what was it that made these fungal spores so hydrophobic? And basically the answer is that these hydrophobin proteins are able to self-assemble into this incredibly robust monolayer that's really hydrophobic. But, and this is where the project sort of stopped with him, was that once these proteins are self-assembled, you just simply could not resolubilize them. People have tried boiling in SDS and boiling in urea, all sorts of things. And it wasn't until someone not in our group discovered that you could resolubilize them by resuspending them in 100% trifluoroacetic acid. Now, hang on a minute. Especially if you come from an entomology background where, your view of proteins is they're like these Goldilocks things, like they're really finicky about activity, 10 millimolar tris pH 8. Oh no, I'm not active under those conditions, thank you very much. So the thought that proteins could be solubilized in TFA and actually still functional, so that you could then polymerize them again and go through this solubilization and polymerization stages any number of times, was pretty mind-blowing at that point. So he gave me some of these purified rodlet preparations, managed to solubilize them. So I was left with small protein that was soluble. I thought, we should be able to get a structure from this. And so I wound up contacting, not directly, but Joel Mackay, who a number of you will know. And you'll know that Joel doesn't do anything by halves. And fortunately for me, he dived into this project because, again, it's one of those ones where, somebody purifies a protein and expects then the structure to be done just like that. How hard can it be? It was hard for a number of reasons I'll get into. But the project evolved more and expanded to involve Margie Sunday, who a lot of you will know as well, and then also Richard Haverkamp, who used to work here a long time ago. I think he's at Massey now, so he's a materials scientist. So there was some, a lot of interesting possibilities what you could do with these proteins. So I'll just flick in the next slide. Sort of got a little bit out of whack here. So I just want you to just look at the images. So this is what the sort of rodlets can look like under various microscopic techniques. So if we look at the left image, so this is a transmission micrograph, which has been shadowed with carbon. So you can see these really sort of this cool rodlet structure. Actually not only got them a Nature paper, but the cover of Nature. Those were the days. And as I said, you could resolubilize these in trifluoroacetic acid, assemble them back into rodlets. This is another TEM image, this is AFM image. And also, interestingly, if you stain them with Congo red, you get the sort of classic red-green birefringence, which you get with amyloid fibres. So, and Margie really picked up on this, and it really suggested that, amyloids didn't necessarily have to be confined to sort of pathological states or instances, but that in nature there was some times when proteins called, or amyloid fibres could form in a sort of functional sense. Anyway, Joel eventually solved the structure. There were a lot of hiccups on the way. Solubility was an issue because it has this sort of preponderance of hydrophobic amino acids, so it was hard to keep soluble. It also had these very large disordered loops. Now, people these days get, more excited about disordered regions than ordered ones. I tell you, this is a pain in the neck at this point. So, but he persevered, got a really nice structure, and developed a model for how they might self-assemble into these rodlets. And we got a really nice set of high impact papers with that, which was pretty cool. Okay, so I should have said that, but as you would have seen, Joel was based at Sydney, so I was visiting Sydney Uni quite a lot, and it let me finish sort of the last bit of my PhD 10 or 20 years later, because my PhD had involved the mode of action of a toxin. So as I said, Robin Mitchell at the DSIR at the time was interested in the structure of really interesting toxic components produced by this pathogen, bacterial pathogen, Pseudomonas syringae. And one of these toxins turned out to be an amazing transition state analogue of its target enzyme. And I linked up with some other people at Sydney University, and they solved the structure of this inhibited complex and showed that it was pretty much as perfect as one could expect, transition state analogue of this enzyme. And again, we got a really nice set of publications with this. And most of the hard work there was done by David Langley. Okay, so while I was sort of diverted on these, we had swapped or moved our main pathogen system from this post-harvest pathogen to another pathogen of apple. And this project was kicked off by Joe Bowen and Kim, who got a Marsden Fund, kick it off. And then I think we moved our foundation money into it. One of the difficult things about working in sort of plant pathogen systems is that, especially at that time, there were very few good model systems. Because as you can imagine, every agricultural plant of importance will have a number of diseases. And the focus on those diseases will depend on where the money's coming from, how important that disease might be in a particular country. So the research was quite fragmented in that sense. It has been drawn together, and now we've got some, really good, robust models, and we understand what's happening, in the bigger picture. But at the time, progress was quite slow. This disease had a lot of positive features. The genetics were much more clear-cut, so there was some characterised resistance genes in apple and avirulence or factor genes in the pathogen. You could cross the pathogen. Apple is much easier it's diploid, so still some tricky things, not a fun fungus always to work with, but it's a much more tractable system. Anyway, this was led primarily by Joe and Kim. Then Carl Messerich did a PhD with us, so I think a lot of you'll know Carl. He, Auckland graduate obviously, but has set up a fantastic lab at Massey University in more recent times. So we were making progress with this system, but then an event occurred that really, especially for me, but for others in the room, changed things completely. So there was this incursion of PSA, which, although it had been present around the rest of the world, still took everyone by surprise here. Just got this picture on the left. Even though this is just an SEM of a colony of PSA, I really like the way that it invokes the sort of an image of the maelstrom that the arrival of this bacterium made. And in only a few months, it reduced, the orchards of particularly Hort 16A to stumps. It was sobering going to growers' meetings. Wasn't anger there, but there was, frustration and sort of a real resigned look that this was it, i.e. the end of kiwi fruit growing. Fortunately, that hasn't been the case, but yeah. So the project that and Eric got the MB bid for this, but it was generally a collaboration with Anna McCann and Paul Rainey as well. So we sequenced a whole bunch of different PSA isolates, and it was the first really big sequencing project. So the pathogen had been around before it came to New Zealand. It was first found in Japan in the 80s and then in Korea. But then there was a global outbreak of this particular clade. It spread around the world very quickly and pretty much wiped out any orchard growing a susceptible Chinensis cultivar. And by sequencing, by this, at the time, quite big sequencing project, we ascertained that the outbreak clade actually wasn't derived from either of the previous outbreaks, but was from some other, at that time, unknown source population. So that was new information. But the other thing that I think surprised me, I can't speak for everyone, but was surprising was just how big and different the accessory genomes were between these, what were, phylogenetically really very closely related isolates. And this is tantamount, obviously, to the amount of horizontal gene transfer that's occurring. But still at the time, I think we're all surprised at how variable these accessory genomes were. I remember thinking once we sequence this isolate, we will find some SNPs, maybe a couple of new genes or not, but actually there was a whole range of different stuff there. Again, got this nice publication out of that. And that led to us, the wider team at Planet Food, being awarded the Prime Minister's Prize. So that was very nice. It was very much a team effort that involved breeders, pathologists. Actually, I'd like to acknowledge two people in this room who helped a lot. Richard did a lot of the legwork. He's not in this photo, but probably deserved to be because he did a lot of the legwork getting that application through. And also Dick Bellamy, because I believe he was a referee. And we all know Dick, when he's positively fired up about something, he is very supportive, and that was the case. So, yeah. Now, this gave us the basis for really drilling down a lot more into just how PSA is causing disease. And I was very lucky to get some money from Bioprotection Aotearoa, hire Jay. It's a bit of a story around that, but so things completely changed once we hired Jay, because he's a complete machine. Oh my goodness, he was doing so much work. So what we were interested in initially was, of the sort of collection of effectors that this pathogen has, which are the really important ones, and which are the ones which, although I've said that Actinidia chinensis is really susceptible, a lot of other species of kiwi fruit are resistant to the pathogen. And that's usually due to the fact that that species will have a resistance gene that's recognising one of the effectors. And the way, a lot of these sort of species complexes work, and I'm talking about Pseudomonas syringae here, is that there's a sort of a huge pan collection of effectors. And for them to cause disease, they've got to get the right team of effectors that enable it to repress the defence response in the plant, gain nutrients, and avoid any resistance genes that might recognise their action. And the team of effectors defines the specificity of the pathogen. So PSA is pathogenic on kiwi fruit, and pretty much only kiwi fruit, because it's gathered the right team of effectors that enables it to colonise kiwi fruit and dodge any resistance genes. A little bit like playing mahjong, if you can imagine that these isolates might be epiphytes on the surface of a plant. They're exchanging effectors using horizontal gene transfer and just need to get the right set to enable it to cause disease. So we were interested in looking at this team of effectors, which were the really important ones, and also which ones trigger resistance in other Actinidia species, because these are the genes that you'd want to breed into the susceptible cultivars. So you'd have some resistant ones instead of horribly susceptible ones that you were spraying with copper all the time. Again, mainly through Jay's efforts, we got some really nice set of publications on this. And Lauren was one, again, superstar student who did tons of work, helped with all these. And she did sort of one last really interesting experiment where Jay had managed to individually knock out all these effectors. So we did this sort of population experiment where we took all these knockouts in individual lines, put them together, and did a sort of a in vitro evolution experiment where we'd infect plants, collect PSA from those plants, and do a whole lot of generational infection experiments. And what we found was that, what Lauren found was that, so I need to go back one, sorry. So the results from this work were that even though PSA's got quite a large number of effectors, there seemed to be only a subset of those that were really crucial. And so the question was, what are the others doing? Are they just hangers-on? Is it just by accident they're there? Or do they have a more subtle role? And so Lauren wanted to test this by knocking each one out individually, making a soup of equal amounts of all of them, infecting plants, and what she found, and then measuring the levels of each mutant. And what she found on susceptible plants was that actually the wild type started quite quickly dominating, which suggested that even by mutating what we thought were some redundant genes, those were still falling out of the population. So it suggested that collectively they all had a significant role in a more of an evolutionary sense than in a sort of immediate single assay, which is what we were doing before. We're also really interested in the question of, when PSA arrived in New Zealand, it was a single clone, and that was now 15 years ago. What does it look like now? And so we've been monitoring orchards and sequencing every couple of years for SNPs and looking for what other elements, what genes might be mutated, etc. And generally what we're finding is that the actual core genome is more or less the same. It's got a very low rate of mutation, but there's lots of horizontal gene transfer coming in. So there's tons of new plasmids that we're seeing. Not 100% sure what they're all doing, but this is sort of a really important piece of work to do for industry, because we kind of need to keeping an eye on this pathogen to make sure it doesn't jump to some of our newer cultivars, which we're relying on to maintain production. Ooh. I'm going to talk about quickly a couple of interesting other topics that we've worked on. So while we know tons of things about horizontal gene transfer in bacteria, we know virtually nothing about horizontal gene transfer in fungi. We know it happens because we can see, for example, identical transposons finding themselves in different fungal genera of different pathogens. And we also see, transposons and little mini chromosomes that are piled full of essentially fungal accessory genes. So they've got no core genes, but they're able to move. People have shown that they can move from different isolate to isolate. So how is this happening? We've got no idea. And one reason that fungi look after themselves and protect themselves from potentially other elements, destructive elements like mini chromosomes or transposons, is that they've got a very strict system for preventing fungal fusion between different isolates. So they've basically set up this genetic system of about 6 or 7 alleles which prevent hyphal fusion, which would be the most logical way these elements would transfer. And Sadia did some really nice work, again based on initial background work by Ross Beaver, and cloned some genes which are involved in this really sophisticated system that fungi have to prevent anastomosis. Identified an interesting set of genes. And what we found, though, is transposons and these dispensable chromosomes are also clustered with a lot of genes that we would think might be involved in fungal anastomosis. So it kind of suggests to us that they may have a role in overcoming, so that these elements, in addition to having particular genes that they might be selected for, also have the machinery to work their way into other isolates. Okay, another busy slide. So the other area that we've become interested in is, how do bacteria recognize self from non-self? Now, I know we know a little bit about quorum sensing, so bacteria can judge if they've got many of their mates around, but how do they know who else is in their niche? And most importantly, they don't want anyone else in their niche, so they're trying to get rid of any other potentially competing bacteria. And I kind of like this story because it starts off with kind of a random observation that leads to something. So Carl Messerich, before he went to Massey, did a one-year postdoc with us, and he made a transposon library out of PSA. In that library, he observed some colony mutants, some rough colony mutants. They're very commonly found in bacteria. Usually, they're mutants in lipopolysaccharide, which is the envelope around bacterium. Lo and behold, when we mapped those transposons, they were in a locus that we thought was involved in lipopolysaccharide biosynthesis. So that's nothing new there. What was kind of strange was that when we compared that locus with some of our really closely related biovars of PSA, we could see that there were at least 2 different alleles, which you could really immediately think why that might be. Shock horror, we did some biochemistry, ran the LPS out on a gel to get this characteristic ladder formation, and the alleles mapped at least the electrophoretic properties of the LPS. Now, just moving aside a little bit, obviously bacteria get viruses, phage, that's all very well characterised, that biology. Phage can either kill their cells immediately or find their way into the chromosome of the host, and then, like a ticking time bomb, lies the cells. But what's not quite so well known is that bacteria have often domesticated these lysogenic phage and turned them into a tool that's actually now useful for them. And they do that by deleting most of the proteins around the head, and you're just left with the contractile element, which can still bind to the outer surface of the cell, which is the lipopolysaccharide, and trigger injection. But in this case, instead of injecting DNA, it's depolarizing the membrane, killing the cell. And what we found was that there's a correlation between the LPS allele and the tail fibers which bind to LPS in the taylosin that a cell would have. Because clearly a cell doesn't want to be targeting itself, right? So it's actually using these taylosins to target competing bacteria. So this is interesting in the case of trying to understand how bacteria recognise self and non-self, and also we believe that this might also be a control method for PSA. Ooh. As you know, I've been involved in teaching for many years here, 2 courses in particular, plant pathology and symbiosis, and 4th year course, plant microbe interactions. I started actually when Dame Alison Stewart was here, gave a few lectures. Didn't have to do that when Kim Plummer was employed, but when she left, came back, and I've contributed to these courses for about 20 years, I guess. Big shout out to the professional teaching fellows who helped me. Credit unduly goes to me for running these courses, but there are other people in the background who do a lot of the work helping me fumble through Canvas and any other obstacle that gets thrown in front of me. So I'm going to have to whip through this because I'm, as usual, hopelessly out of time. So I just want to sort of almost finish with some of the things that really sort of blow my mind about the sophistication that pathogens have for causing disease. And, as more and more research goes on, the more sort of mind-blowing I find this. So I said pathogens, effectors, their role is to repress the defence response. And there's been lots of work in that area. More recently, people have found a number of effectors have a role in reprogramming the endomembrane system within a plant cell to the advantage of a pathogen. And just here's an example of that. So this is a fungal pathogen of rice. So imagine you're looking top down on a rice leaf. Here are some rice epithelial cells, or here's a epithelial cell. It's been infected by this fungus. The fungus has been transformed with 2 GFPs linked to 2 different sort of effector proteins. One localizes in the space between the fungal wall and the plant cell membrane. So the first thing that actually happens here, one thing to note is that the fungus, when it enters the cell, it doesn't pop the plasma membrane. Imagine you've got a sort of a balloon that's been poorly inflated and you push your hand into it. So that's basically what the fungus is doing. It doesn't pop that membrane. What it's done is it's secreted some effectors which have completely changed the internal dynamics of the endomembrane system in that plant cell. So it's essentially moving the vacuolar membrane into this membrane which surrounds the invading hyphae. And the plasma membrane is sort of, it's unpleasant for a fungus. It produces reactive oxygen. It'd be like wearing a hairy shirt or something like that. But it manages to persuade the plant to have this much smoother, sort of satin, silky type membrane, which is much more comfortable for it. And so it's secreted some effectors into that space, but it's secreted also effectors which actually are acting within the plant. So it's secreted them through this plant-generated body. For visualization purposes, they've put a nuclear tag on it, so it goes to the nucleus, doesn't usually go there, but that's just so that you can see this. You can see in this next image that these effectors are found or finding their way into other cells. So in the middle one, you can see the effectors have moved into these other cells. So they'll be doing their job, repressing the defense response, going to each cell, switching off, finding the defense response switch, switching it off. So that makes it easier then for the fungus to colonize those cells. The next amazing thing is they move from cell to cell. And they do this by finding, remember, they don't pop a membrane here. They find the plasmodesmata, squeeze in sort of Houdini-like, and manage to go from cell to cell by doing this constriction and then just popping back out again. Sorry, I find that amazing, but anyway. This is annoying. Right, so the next amazing thing I find is these are a set of effectors called TAL effectors. They're transcription activators. They're produced by a particular genus of plant pathogenic bacterium, and it uses these to switch on genes to its own advantage. So most of the work has been on sweet genes in the plant. So these are sucrose transporters. So it's switching on sucrose transporters to basically feed itself. But these proteins are repeat proteins that can recognize particular bases in a double helix. And so they're evolved to bind to particular promoter regions, activate those genes. And for a while, these were used for doing gene knockouts in plants, because as you can imagine, you can just redesign these proteins whatever way you want to recognize a particular motif, and then you can engineer a nuclease in the middle and create a double-stranded break and a mutation. But of course, this all got blown away with CRISPR. Although, given the messy IP with CRISPR, these guys may come back into... Yeah, I'm running out of time, so I'm going to skip this. I just really want to go through some of my students, because obviously you can't get anywhere without high-quality students. I've had some fantastic PhD students starting off some time ago. So these are co-supervisions and main supervisions. Joe Wynn, he's now in one of the best top plant microbe interactions team in the UK. Robert Weinfeld did some of the AFM work on hydrophobins. Carl Messerich, Sadia did the fantastic work on vegetative incompatibility and botrytis. Erin, superstar PhD student, she's now doing a postdoc with Murray Grant in Warwick. Lauren, fantastic student, she's postdocing in Toronto. Didn't talk so much about Hoop's work or Liz's. Liz is doing postdoc in Kiel. Brogan is still doing a PhD, and some other people I didn't have pickies of, unfortunately. But as I said, can't get anywhere without quality students. Also, you're not going to get very far without funding. So I've had a big variety of funding sources, starting really with some internal funding, but also various iterations of MB, Royal Society, being PI on a couple of Marsden grants. Probably the big game changer for me, though, was getting bioprotection money, which allowed me to have a postdoc. I can't see how I'd be in this position without that. I think I'd still be languishing at AP without that postdoc work. Also, thank the university, mainly through scholarships for my PhD students. More recently, I've been getting money from Zespri and KVH, so I've had a range of money from industry to pure science with the Marsden. Just a couple of final thoughts. I can't see how we can make the most of the impacts from this work without engineered plants. We've really got to be able to move resistance genes, even if it's only within a genus, would make a huge difference. So in this image, it's from a group that have put 3 resistance genes to late blight in potato, which is the Irish famine disease. So it's still a big problem throughout the world. A lot of chemicals are used to control this oomycete. But by pyramiding 3 resistance genes, it makes it almost impossible for the pathogen to evolve resistance. So in this plot, we've got resistant plants at the top, susceptible ones at the bottom. And then last thing I want to say is obviously the Joint Grad School, I think has been a fantastic innovation. I've benefited a lot from it. And I'm obviously, hoping that it can further evolve with the new pros. But I think, more needs to be done. A lot of it is sort of almost half in kind. It really needs to have it needs to have additional funding for PhD scholarships. And one of the things that Robin and I are really big on is creating a much smoother pathway from undergraduate to PhD to employment. And at the moment, that path is still quite bumpy. So we've worked really hard to get industry to have prizes for some of our top students. Where we can, we then try to find summer studentships for them, then PhD scholarships. And I feel really strongly we need to have a much clearer pathway without any gaps in it. And you can have all the money in the world and great students, but you've really got to have a nice, vibrant working spot, which I'm very fortunate that I do. I've always had really good colleagues and managers. So one of the fun things that Erin organised for our floor was Funky Shirt Friday, and although it's sort of lost a little bit of oomph, but it just gives you, tells you of the fantastic vibe that we do have, and it's fantastic. It means that every day wake up and want to go to work, which is great. That's it. Thank you.